Okay, let's go. Finish chapter 15. Let's look about where we left off. We just got done talking about charging up a tRNA. And now we are on the ribosome. So the ribosome itself will have three major active sites: the P, the A, and the E site. So the P site, basically, this is where your growing peptide is. So P for peptide. And so this is the area where the tRNA that's in there will be holding on to the long chain that you're working on. In the A site, this is kind of like an empty chamber. I call this the arrival site, right? This is where the new codon in line is going to be read. So how you choose your next amino acid in the sequence, and that tRNA carrying the next amino acid that you're going to be adding to it will arrive in the A site. And then E is the exit site or the ejection site. So basically, once you deliver your amino acid, get a little bit of a shift over. You have three tRNAs. One's holding the growing polypeptide chain. The other one's holding the new amino acid that's coming in to add to the growing chain. What does the third tRNA have? Nothing, because it already delivered its amino acid. So it has nothing now. It's an empty tRNA. So basically what it does is it gets ejected, kicked out, so that it can go get recharged, which was the topic that we were talking about on this slide, right? So you as a tRNA are just like a taxi. You basically go grab an amino acid, go deliver it, get kicked out, go grab another amino acid, deliver it, go get kicked out. So very similar probably to the life of a taxi driver. When you take a look at the ribosome, this is what it looks like. So here you can see you've got your large subunit and your small subunit. Your large subunit is actually where the EPA sites are going to be. Your small subunit is kind of there to sort of bind to your mRNA and hang on to that. So this is the one that kind of hangs on to your code, if you will, that you're reading off. And then basically you sort of read off your genetic code. Now notice what happens though in these sites. You have in these sites for three base pairs, right? This is the reason why a single codon, three-letter word, is about the size, just the right size to fit into one of these active sites. Okay, so this is your ribosome. This is actually a real ribosome that we finally kind of did the structure for. We had a lot of celebration over this one for a long time. Many careers involved in this one. So we were very excited, very happy when this finally came along. The one thing I do like about this particular slide to sort of drive home the point is what the ribosome actually does. The ribosome's job, like its catalytic job, is simply a transfer. That's what it does. And in the process of that transfer, it creates a peptide bond. And the enzyme activity we refer to as peptidal transferase. So essentially what it does is it transfers the incoming amino acid to the growing chain. That's what it does for a living, and that's all it does. Notice it also bears reminding when it comes to quality control, right? Making sure that the correct amino acid is on the correct tRNA or that the correct amino acid is being delivered. The ribosome has nothing to do with that, right? So it does not sit down there and kind of check you down and say like, oh, whoa, wait, I think you're carrying the wrong amino acid. It doesn't do any of that. All it does is take what you give it and then add it to the chain. That's all it does. So you could give it a completely disastrous amino acid and it'll just grab it and add it to the chain. The quality control, if you recall, is actually all resting on the shoulders of the aminoacyl tRNA synthetase, which was the charging enzyme whose job it is to actually attach the amino acid to the tRNA in the first place. Okay, that's where all your quality control comes in. Okay, let's go ahead and take a look then at translation in prokaryotes. So what do we need in order to initiate? Now, the first thing, obviously, because we have this three-step mechanism, right? Initiation, elongation, termination. So what do we need in prokaryotes to essentially create an initiation complex? Well, you need three things. First of all, you need an initiator tRNA, right? What does that mean? This is the first one in line. This is your starter. And typically this initiator tRNA is all the same in bacteria. It's basically carrying an N form of methionine. If you recall, methionine is one of the amino acids. In this case, it's kind of like a slight modification. It's got a formal group on it. But that's something that's a bacterial thing. So still a methionine. The other thing you obviously need is a small ribosomal subunit, and then of course you need something to read, right? Your mRNA strand. So this is what you get. So you got to assemble them. Okay, so it's just kind of like cracking open, like some sort of a delivery from Amazon, some assembly required. You basically have this bag of parts, and hopefully you got some instructions. Yeah, we have right now this bag of parts. Well, how do we do that? First of all, we got to figure out how to bind the mRNA to the small subunit of the ribosome, right? Because if you can't hang on to your mRNA, you're not going to be able to read it. Now, how do you do that for your bacteria? It's sequence driven. So in your mRNA, you have a little stretch of sequence called the ribosome binding sequence. And it literally is kind of like a sequence signal that will basically allow the small subunit to bind to that particular area of the mRNA. So once you bind your mRNA through your RBS to your small subunit, then all you have to do is position your initiator tRNA to that mRNA. And then once you get all that together, you're going to cap it with the large subunit, and then away you go. Okay, so let's take a look at it. Right? This is truly the case where a picture is worth 1,000 words. So this is a type of chapter where that is certainly true. Yeah. No, it's a totally different sequence. It's not like the 5' cap or anything like that. That's only a eukaryotic thing. This is a prokaryotic thing, right? We will talk about that, however, in just a second. Right, so first of all, notice here's your start codon. Notice where it's positioned, right? So it's kind of positioned downstream a little bit. So what you have up here is kind of like a little region that doesn't actually get translated. This is actually something that's true in the architecture of mRNA. There's actually like little leader pieces. If you recall, the open reading frame that we talked about before is the part that becomes protein. That's actually the part that becomes translated. That's where your AUG is. But between your start codon and your stop codon, there's more to your transcript, right? So there's like a 5' piece of it, a little piece that doesn't become part of the protein. We call that the 5' untranslated region. And there's a 3' part of it, right? That's where that poly A tail is going to be, right? There's a 3' untranslated region. Typically in mRNA, those are usually have regulatory functions. A good example is this guy right here. So this would be your 5' untranslated region. In this particular case, that's where your RBS is. And so this kind of creates your little binding signal. So it's going to bind to the small subunit. One thing I want you to notice though, is notice that you have 3 pieces that need to come together, right? But you notice what's happening. They don't just randomly drift around bumping into each other and hoping you kind of get the magic, combination of all 3 of these guys bumping into each other at the same time. Notice nothing's that random. When these things are coming together, they're being shuttled. So if you take a look at this, you have a whole host of initiation factors, proteins, whose job it is to make sure that everybody is where they need to be. So there's an initiating factor that basically its job is to grab onto the mRNA and to get it to the small subunit so it can bind there through its RBS. Notice there's also an initiation factor whose job it is to grab onto the tRNA and to move the tRNA to its correct position. I like to think of these initiation factors as sort of like stagehands in a good theatrical presentation, right? If the stagehands are doing their job, you never notice them. But if you're an actor on stage, you'll notice that they're critical, right? Because the stagehands are the ones that are helping you make sure that you find your mark. You're able to look at a stage from the top, which you never see from the audience because you're looking at it from the front. But if you look at it from the top, you'll actually notice there's all these marks and things like that on the stage, because they're marking out where different characters and different actors have to be when their scene starts, right? And making sure that those marks are in the right positions and that the actors are on their marks when the scene starts are the stagehands. That's like the initiation factors. So the initiation factors are basically busy shuttling all of these pieces together to make sure that everybody is in the correct place at the correct time. And then once everybody gets to the correct place at the correct time, you notice you basically have your mRNA now fully bound by the small subunit, and you have your initiation tRNA now locked on to your start codon with what? Your N-formal methionine as your first amino acid in line. Now you're ready to roll, aren't you? Basically cap it, and when you cap it, the P site, one in the middle, will basically have the growing chain. In this case, the growing chain is 1, but that basically now is in the P site. Notice your A site is empty, but it's got the next codon in line for you to read. And then the E side, of course, is empty because, well, there's nothing there, right? So you've already spent that one. So now you're ready. This is basically the events of initiation and translation. As it turns out, initiation of eukaryotes is very similar. Right? When we basically took a look at this, we're like, oh, hey, wait a minute. This is pretty much the same, right? Most of this is the same, with the exception of a few minor details. That initiating amino acid instead of N is just straight methionine. So there's no modification in eukaryotes. We have a little bit more going on in our initiation complex, as you probably well expect, right? So after looking at all those transcription factors in transcription, it probably doesn't come as a surprise that eukaryotes are always more complicated in nature, right? So eukaryotes are sort of like teenagers, whereas prokaryotes are like little kids. Yeah, little kids are fairly simple and easy to please. Teenagers help us all, right? They're all over the place and they're considerably more complicated. Nobody knows what they're thinking or doing half the time, including them. Just the whole world is waiting patiently for them to grow up. And that's basically teenage existence in a nutshell. Right? Because even the teenagers themselves are waiting patiently to grow up. And sometimes not so patient. So here's the problem though. We don't have an RBS in eukaryotes. So we don't have that sequence to bind to a small subunit. So how do we pull it off? Well, actually what we do is we use our 5' cap. So instead of an RBS, we substitute that with our 5' cap. We've already got it. We need to have it, right, in order to protect our 5' end from degradation. So we just basically utilize that as our binding strategy to our small subunit. So initiation otherwise is pretty much the same. Now, what about elongation? Well, in elongation, actually, there's a few steps in elongation. And elongation has a little bit more complexity. So we're going to take a look at that stepwise. But again, I want to drive home the point that elongation, just like initiation, isn't just magically happening randomly, right? You've got elongation factors, right? Your stagehands that are behind the scenes, making sure that everybody is where they need to be and everything is happening correctly. One of these stagehands, one of the more important ones, is EF2. So EF for elongation factor 2. And so this one basically is responsible for taking tRNA and making sure that it arrives at the A site. That's what that one does. And then once you get to that particular site, then you're ready to go ahead and add that amino acid to the growing chain. Elongation happens in a 3-step cycle in its own right. So we have this 3-step cycle within a 3-step cycle, right? So within the initiation, elongation, termination, we also have in elongation a 3-step system. What's the first part of elongation? It's codon recognition. Here, the tRNA anticodon pairs with the codon on the mRNA, and also delivering the correct amino acid. So here's an example. So here you can see you've got your open codon down here that is ready to be read. Your elongation factor, EF2, is going to bring that correct tRNA associated with that codon into position. And it's going to lock into position through complementarity. And then once it's locked in, then the EF2 leaves and you have successfully managed codon recognition. Okay. The second step is peptide transfer. And in this particular situation, the peptide chain moves behind the new amino acid. So what's going to happen is this growing chain right here, which is now 2, is basically going to switch and add to or behind the new amino acid that came in. Now, this is kind of important, right? Because when you're making the chain, it's like growing a line. But the problem is whenever you show up to a line, like for instance, concert tickets or something of that nature, where do you go when you see a big long line? Yeah, you go to the back. What happens if you go to the front? If you're lucky, you could get beat up depending on which concert you're going to, right? So you go to the back of the line, right? But in this particular case, it's actually just, it's different. When you do peptide transfer, you don't go to the back of the line. The back of the line goes behind you. So the new amino acid that shows up, the line jumps behind you. So the new amino acid is now the one that's at the front, right? So that's the transfer that happens. But now what do you have at the end of peptide transfer? You've got a problem here, don't you? Because now the tRNA with your growing chain is in the wrong site, isn't it? It's in the A site. And not only that, but the one that was in the P site is empty. So where does it belong? It belongs in the P site, right? That's where the empty one goes. So then that basically gives us our third and final step. That's translocation. So the mRNA threads through to expose the new codon. So what's going to happen in translocation is you're basically going to pull your mRNA this direction. What that does, it's going to shift the empty tRNA into the E site where it belongs, right? It's going to shift the tRNA with the growing chain into the P site where it belongs, and it's going to leave open the A site with your new codon that needs to be read, the next one in line. Of course, in the E site, it's empty. We call it the ejection site for a reason. So once you're empty, you get kicked out of the ribosome, and then basically you're going to go get recharged. Yep. Go back to the amino acid tRNA synthetase, go pick up another amino acid, and then come back and do it again. And then at that particular point, we're right back where we started with codon recognition. Only in this case, we're recognizing the next codon in line. So let's take a closer look at peptide transfer. This is that step 2 in elongation. So here you can see you've got your new amino acid coming in. This is the original amino acid that was already there. What actually happens when this occurs is that the nitrogen will actually attack the carbon and basically you're going to form a bond between those two. When that happens, that's going to break this bond and it's going to cause this amino acid to jump over here. And so then what you get is a growing chain, but when you look closely at it, this is going to be your amino group. And down here, that's being held on to the tRNA, is your carboxyl group. Meaning that as the chain grows, every protein is going to have what's called an N-terminus, where your amino group is hanging out. It's going to have a C-terminus, where your carboxy group is hanging out. And then you're going to have basically your linear chain. Okay. Now, generally speaking, because the green amino acid was there first, that's methionine, right? Then we typically start numbering from the N-terminus. N-terminus represents the front of the line. So in reality, you're not really moving a line behind you, are you? You're moving the line in front of you is what you're actually doing. So you're still at the end of the line. It's just that instead of you getting behind the line gets in front of you. That makes sense? That's kind of what's happening here. And then basically what happens is you do translocation and you do it all over again. Now, there is something that we noticed and we discovered in this translation process that's kind of like a little bit of a weirdness, if you will. Generally speaking, when you're looking at like an anticodon, was that the first sites have very stringent base pairing to them. So that is to say, this has to be A. Same thing with this one. This has to be A. These two are very stringent, but this one, not as stringent. So this one normally would be U, but maybe it could be G, maybe it could be C. So there's some options. So there's some flexibility in this last one. Because there's some flexibility in that last one, it means that basically you can have different tRNAs encoded. Right, so you can kind of get better, more flexible pairing. And so that wobble is gives you a little bit of breathing room, essentially. So it's kind of a weirdness. So notice what we have is not a hard and fast code. We have a code, but then if we had a really strict letter to the law kind of a code, it would create a lot of restriction. And then we would have to basically create like a lot of tRNAs. And then that would be really expensive, right? So that would be kind of energetically expensive for us to do. So what this does by creating a little bit of flexibility in this, it means we can still build a functional protein, but we can also save some money and not have to make as many tRNAs to still get the same effect. Remember, the cell doesn't want to buy the Bentley. The cell just wants to buy a functional car. It wants to save all the extra money for other things, right? So the cell is one of the best economists out there. There's not a lot of frivolous spending at the cell level. Termination, of course, you just basically motor along then, going through your codon recognition, peptide transfer, translocation, and you just keep on doing this until what enters into your termination site is your stop codon. So what happens basically when you hit a stop codon? Well, you actually have something else that binds there, a releasing factor that binds into your stop codon. So here you can see a stop codon now is entered into the A site. But now instead of a tRNA tethered to an amino acid, you've got another protein called a release factor, which is basically going to bind to that codon. And there's nothing there, right? So now all of a sudden you get codon recognition, but it stalls out at the peptide transfer state. And it's like the ribosome's like, so where's my amino acid, right? So where do I put my growing chain? Well, what happens is when you stall out like that, eventually there'll be a bit of a pause, but usually almost always the end result of that kind of a stall out or a pause for any kind of a complex like this is dissociation. Basically, it's going to fall apart. And what it does, it's going to release your polypeptide, it's going to release the releasing factor, the tRNA, and you're going to dissociate your ribosome. And guess what? You go right back to initiation. The rest for the weary, just go and grab another mRNA that needs to be translated, assemble yourself an initiation, form your initiation complex, go through translation, elongation until you hit your stop codon, and then do it all over again. And you do it over and over and over and over and over and over. And that's essentially what ribosomes are doing. That's all they're doing, nonstop. So, now what if you have, say, for instance, a protein that's going to be targeted in a particular area? Like, so for instance, let's take a good example would be like proteins of the rough ER, right? There's a good example, right? These are ribosomes and proteins that are actually located in the rough ER. Why? Because they're probably going to be secreted. Or now we had 3 different proteins that were involved there. One of them was secretion. We're just going to use the secretion one as an example, right? So you want to be able to localize this and sort of locate this to that particular place. And so we can do that. But the way we do that is what's called an SRP, right? A signal recognition particle. And you notice we saw this one before, didn't we? Right? This was part of the RNA group. But the signal recognition particle is a piece of sequence that acts like a zip code. Telling you where to go. Now, of course, if this is a particle, right, a piece of the polypeptide is protein, then that means there had to be a code for it, right? So this was something that was also on the mRNA, and that's kind of what we're talking about is the mRNA piece. We were talking about that earlier. But basically, this is a signal sequence that will allow ribosomes to dock their to their rough ER, right? So a lot of times students be like, well, wait a minute, how does the ribosomes on the rough ER know to go to the rough ER, right? And what's that process? Well, this is basically that process, because once you actually dock to the rough ER, then you get that secretory pathway. Here's what it looks like. So here you can see you've got a little bit of an RNA code in there. So this is your signal that's part of your polypeptide. Then your signal recognition particle will actually come in, and it'll recognize this little zip code kind of tag that's on your polypeptide, and it'll bind to it. So it'll bind not only to this, so here's your binding spot to that, but it'll inject itself into the actual ribosome. So this piece right here actually gets injected into the ribosome, inserted there. So now what you have is sort of like a shuttle, right? Now, as it turns out, this SRP, which is bound to your polypeptide, will also have a receptor, and I'm going to blow this up a little bit, that's in the ER. So if you take a look at this little brown piece right here, this is a receptor whose job it is to recognize the SRP. So it's kind of, if you look at it, it's kind of like a yin-yang thing, right? So they're kind of like two halves of a whole. So when the SRP finds this little receptor in the rough ER, then it basically locks onto it, it connects into it, and then that inserts the SRP, the other end of the SRP, into the membrane of the rough ER. But notice this little receptor isn't alone. It's actually tethered to and associated with a channel that will act as sort of the tunnel through which now you can thread your polypeptide. So then your polypeptide gets injected into the lumen of the rough ER, and then it goes through protein folding. And then from there, we're back to chapter 5, right? Or chapter 4 rather, right? With secretory pathway. So what we did is actually we kind of connected a piece to the beginning of that secretory pathway discussion that we saw in chapter 5. But the same thing would be true if you're docking a ribosome to anywhere else. So if you have a ribosome that's tethered to the nucleus, then you would have an SRP that would recognize the nucleus and would do exactly the same thing. It would basically dock itself to the nucleus. So by doing this, then you can localize your protein production, and you can associate your ribosomes with specific organelles. So here's an example of one of these protein channels you can see up there with its little receptor hanging out by it, waiting for an SRP to recognize it. Okay. So let's wrap it all up, shall we? So what we've done so far. So transcription, we start off with an RNA polymerase, so we'll go in and we'll unwind a piece of DNA. You're only basically making a copy of DNA from one template strand. The other one is not going to be utilized. This oftentimes is something that confuses students, because sometimes you'll hear two different terms used with the single-stranded DNA and transcription. You'll hear the term template and coding, right? The coding strand and the template strand. The one that's used for the information is the template strand, right? Because that's the one you're making your RNA transcript from. The one that is not used is called the coding strand. A lot of times this throws students off because they're like, well, wait a minute, why is it coding strand? Well, you're not using it for coding. Well, because think about it. The transcript you're making is complementary to your template strand, yes? With the exception of U's instead of T's. This coding strand is complementary to the template strand, yes? Only has T's instead of U's. So the coding strand and your transcript are essentially the same, with the exception of U's in the transcript versus T's in the DNA. So the reason why they call the alternative strand the coding strand is because literally it looks exactly like your transcript, but you swap out T's for U's. And that kind of throws students off. So you basically go through your transcription, and then once you transcribe your transcript, remember it's premature at this point. So it's pre-mRNA. And at this point, you got to modify your RNA. You got to cap it, you got to tail it, and you got to splice it. And then ultimately, you're going to create your open reading frame. Now, in your mature RNA, remember the pieces that you have obviously are your 5' cap, your 3' tail. But inside this region here, remember, this is where you get to those untranslated regions. You're going to have a 5' untranslated region. And then you're going to have your start codon, right? Your AUG. And then you're going to have your 3' untranslated region and your poly A tail. 5' cap. So these are those regulatory regions that oftentimes we'll use when processing and working with mRNA. Okay, that's where we found the RBS, for instance, in prokaryotes. So once we process it, then spliced out, we've got an open reading frame. So there's our stop codon there. Then we have to transport it out of the nucleus, where we're going to assemble it into a ribosome. So that's our initiation. Right, and so then once we get to initiation, then we're going to have our 3-step elongation, right? So here is our codon recognition state, and then we have our peptide transfer state, and then we have our translocation state, right? So our 3 steps of elongation. So this is all elongation of translation. Of course, our termination is easy. You run into a stop codon and it just all falls apart. So that's a good chunk of the chapter, by the way, all in one fell swoop. So the smart money would say, pay attention to that little rundown, right? Because that pulls it all together. Yeah.
What is the coupling? So with AUG, what was the other one that translated to it and messed up on?
Not sure what part you're referring to.
I'm trying to. When we did the translation of the last Thursday, it's the same. It's split on
Are you talking about alternative splicing?
The coding chain and the template.
Oh, the coding versus template. This one.
Yeah, I'm just trying to show that.
Yeah, so the best way to remember this one, you can actually see it. And this is the reason why I say, like, when we did the genetic code table, practice that because it helps to illustrate that. You can see it visually, right? So for instance, this guy right here, this top one is our template, right? Because that's the one we're using in reference for our transcript, which is this guy right here, which makes this one down here the complement to it, the coding sequence. Why? Because if you take a look at it, it's exactly the same with the exception of U's instead of T's. And that's because we're doing the same thing. It's a complement, but we're making it a RNA complement instead of a DNA complement.
I thought so.
Is that?
Yeah, I just wanted to
Yeah.
go ahead and make sure that I could.
But you'll see that terminology, like you'll see coding versus template strand, and a lot of times that throws students off, and they're like, "Wait a minute, what is that?" And that's what we're talking about. So now the important thing is when you take a look at this, and this is kind of, so we're talking about gene expression, right? This basically is sort of gene expression in a nutshell. But the reason why we were interested in gene expression, and this is totally a eukaryotic thing, right? Because it's like, okay, great. So you get gene expression, you get all these complexities in this. I mean, just the RNA processing alone, which is only a eukaryotic thing. I mean, there's a lot going on there. I mean, I saw your glassy-eyed look when we were going over alternative splicing. That's to be expected. But I saw the glassy-eyed look even more when we went over like codon recognition, peptide transfer, translocation, and all that sort of stuff during translational elongation. It just seems like this could be more efficient. Doesn't it seem like it's gonna be more efficient? Like it's unnecessarily complicated. It's like, why? I mean, couldn't we just do this like in one step and just like be done with it? Well, why do you have multiple steps? Remember, you carry out for teenagers. They think they have a good reason for being as complicated as they are. Now, granted, most of the adult world disagrees with them, but that's okay. Right? So when you're talking about a eukaryote, the reason why you do things is very intentional. Remember, one of the big messages in all of biology is that doing stuff is costly. And the one thing you don't have in biology is discretionary anything. So there's no discretionary energy, money, right? There's no discretionary time. There's nothing discretionary, right? So if it's not productive and purposeful and functional, then it is gone, right? It's brutal in that sense. Kind of like Deion Sanders with the CU Buffs roster. Yeah, I have to admit, that's a new one. I've never actually quite seen that, right? Where half of the team hits the transfer portal. But anyway, so here's the thing, right? You don't just do complexity just for kicks and grins, because complexity is costly. So you know that when you're doing something and having multiple steps in there, you're not just being difficult, right? There is a very, very valuable purpose, right? We learned about this one, didn't we? When we first started that discussion about sexual reproduction, we said for all the headaches that sexual reproduction gives us, there has got to be a titanic upside. And we found what that upside was, that basically justified all of the expense. The same thing's true here. When you've got this, I mean, mechanism upon mechanism upon mechanism and process upon process, then there's got to be a titanic upside to it. What is that? With gene expression comes gene regulation. And remember, always think of it in terms of regulation. Think of it as like theatrical lighting. I love theater, not so much for the actual productions themselves, but to see what, like, the people that you don't talk about, what they do with the set, like the set designers, the lighting people. If you've ever gone into theater, you know that lighting is not just lighting. It's not just a light switch, you don't turn it on. If you look at the lighting system of most theaters, I mean, it is, you need like an engineering degree to figure out what's going on there. There's actually entire liberal arts degrees just in theatrical lighting. Why? Because you can create an amazing set of optical illusions with just being able to sort of mess with and attenuate light. And you can create like more tension and more drama with light in a theater than you can with the actual production itself. The dirty secret of theatrical performances is actually most of the talent isn't actually in the person writing the production. That's the one who gets all of the accolades. But in reality, the unsung heroes are all the stage people, the stage effects, the lighting in particular. Lighting for theatrical performances is a lot like music is for horror movies. You get scared without even having to watch the movie because you can hear it in the music. Lighting does the same thing. But guess what? A good lighting panel, every single light has its own control and its own attenuation. So you can create an infinite number of possibilities lighting wise. I've seen some amazing optical illusions created in theatrical lighting, where it's like you're sitting there looking at it and you're like, what am I looking at? It's like, and you know it's not there, but it's like you can totally see it. And it's like they're trying tricks on your mind with like a lighting optical illusion. And then of course you have to go see it again and pay more money, which is the point, right? Gene regulation is like theatrical lighting strategy to gene expressions, the actual theatrical lights themselves. Okay. Now, what does this mean? That means that every single step you have genetic control. So how do you express yourself as a theatrical lighting person? Well, there's different levels of control that you can do. In genes, you can do transcriptional control. So if you're controlling genes at this level, whether they're turning on or off, this will be considered transcriptional regulation. And we talked a lot about that, right? Especially with like cell division genes. You either turn them on or you turn them off. However, it's not just about turning them on and off. It can also be like a dimmer switch. It's like, do you attenuate it? Do you slightly increase its strength or do you decrease its strength and get attenuated shades of gray in there? So that's transcriptional regulation. But what about if you transcribe it, but then you do things like alternative splicing? Alternative splicing would be post-transcriptional regulation. So that's an example of modifying your expression pattern, but after you transcribe it. So transcription is fairly straightforward, but now you're basically altering the way you're transcribed or the way you're actually expressed by altering your splicing patterns, right? So that would be post-transcriptional regulation. But then what if you go through that, then you kind of get into translational regulation. Well, believe it or not, once the mRNA is made, you're not obligated to translate it, like at all. Some mRNAs you'll actually sequester, and you'll say like, I'm going to make the mRNA, but no, you wait. So I'm going to maybe fold you up, like into a hairpin or something like that, so you're inaccessible. So you're off. And then once I'm ready for you, then I'm going to open you up. Or what you can do is you can dedicate more ribosomes to that particular spot, kind of using your signal. So if that's true, if that's what you're doing, then you can control translation. This is translational control of your expression. If you make it, doesn't mean you have to translate it. You can have mRNA floating around for whenever you need it, right? Why would that be a benefit? Well, that'd be kind of nice if you need to have a quick response quickly if a certain circumstance arises, and you don't want to take the time to have to go back and transcribe it all, right? You want it to be ready to go when it's ready to go. So in that particular case, what you have is you have a whole bunch of mRNAs that are just sort of hanging out. And once your circumstance occurs, then it's like, okay, great. I've got the mRNA ready to go. So let's fire this off. Okay. Then what happens after that, when you have to make your protein, then you can modify it. With things like carbohydrates, like glycoproteins, for instance. This would be a type of post-translational. Computer away. So you can see everything in a box are places where you can control expression. Now think about, now that's just in the production of a protein. Now think about all the little controllers that you can do and all the nuances and the shades that you can create, the hues of function that you can create with these major regulation spots. You can create nearly an infinite number of effects, can't you? A gene can literally almost have an infinite profile of expression. It's like taking a color and having nearly an infinite number of hues that you can create with that. And guess what? Why are you doing this? What's the upside of all this? This is all costly, right? So why are you doing all this? Why? Because it's just the same, it's the exact same reason that we have genetics. Not only do you want massive variation in your genetics in order to be able to adapt and survive to a changing environment, but even with that genetics, you want to have a near infinite capacity to variably express your genetics, which is already variable. in order to create another set of infinite outcomes in terms of expression patterns and profiles. The more of which you have, the more likely you're going to be able to adapt to a changing environment. So you notice what we have is we have variation upon variation, upon variation, upon variation. And the more variation we have, the more likely we're going to survive anything the environment can throw at us. So this is for you table people, it's kind of like an organizational thing, right? This is just kind of outlining some of the differences between prokaryotes and eukaryotes. For instance, introns, prokaryotes don't have any introns. The genes, right? The operonic nature of prokaryotes. We have one gene per mRNA, so that's idea that we talked about. The site of transcription, of course, in eukaryotes, it's in the nucleus. That is transcription in the nucleus. Translation is outside the nucleus. Your initiation codon is the AUG, is your start codon. The same thing is also true for us, only we use a 5' cap instead of a ribosome binding sequence to get our mRNA bound. And of course, modifications, right? So there's no modifications of prokaryotes. They don't have introns. They don't need a 5' cap. They don't need a 3' tail. We need all of that. Okay. You screw it all up, right? That's always an important one. So the expression of a gene, how do you screw that one up? We talked a little bit about this, but we want to kind of fill this one out a little bit. And then we'll talk about mutations. So one of the biggest things you can do essentially to alter the expression of a gene is obviously to alter its sequence. And if you alter its sequence, it's what's referred to as a mutation, right? So it's altered sequence. And generally speaking, there's a couple of ways you can do that. The first and simplest one is what's called a point mutation. This is a single base change in the DNA sequence. And you have a couple of different types. The first one is what's called a silent mutation. This is where a single base change does not result in a change amino acid. Remember, we saw from the genetic code that some amino acids have multiple codons that will lead to them, like lysine, for instance, has 4 different codons that will basically encode for it. Well, if you mutate from one of those codons to the other, you didn't change the protein. So there's no net effect at the protein level. So you don't really, that's why they call it silent. You're not going to notice any change there. However, what happens if you do notice a change? Well, those are called missense mutations. This is basically where your base pair change is going to change the amino acid assignment. You have 2 different types. Transitions are going to be purine to purine changes. So that's one double dog dish to another double dog dish, or pyrimidine to pyrimidine changes. That's one taxi to another taxi, so TC, for instance. Those are transitions. One going from the same one base to the same base. And then transversions are when you mix that up. So when you go from a double dog dish to a single dog dish, so it's a bit of a more of a disturbance structurally. So if you go to like A to T, so that's your change. This one is more like an A to G. That's a transition. Transition is purine to purine in this case. This is purine to pyrimidine. A little bit more disturbing, right? Because you go from that double dog dish structure to the single dog dish structure. If you recall, the way that purines and pyrimidines line up with each other in base pairs, the fact that one's a double dog dish and the other's a single dog dish kind of creates that structural sort of a hitch in the giddy up that causes that sort of like weird little minor major groove structure that we see in DNA. If you start to disrupt that, then it's going to foul up that basic geometry of DNA. It's going to cause a little bit of a weirdness in that. Not for that, but this will actually result in a different amino acid, which could be catastrophic. The worst of them all, and this is basically an increasing severity of the mutation, is what's called a nonsense mutation. This is basically where it introduces a premature stop. And what this will do is it'll truncate the protein. So for instance, if your single base pair mutation turns it into a stop codon, then you've terminated that protein prematurely, and that's called a nonsense mutation. So for instance, if you have a UUA and this gets mutated to a UGA, then that becomes a stop codon. And then that abruptly terminates that. So this is what it looks like. So here's your normal, and here you can see they're using the coding and the template language here. So here's your mRNA. You can see you've got AUG, CCU, UAU, CGC, UGA is your stop. And then what they're going to do is they're going to do a series of changes here. So the first thing they're going to do is this CG one right there. So this is going to be a silent. So if you notice, it's going to go from CCU to CCA. Both of those encode a proline. So even though you get a mutation, you actually don't get anything in the protein. It's still methionine, proline, threonine, arginine. Okay, so nothing happened there. It's a silent mutation. Now the next one they're going to work on is an AT switch. So in this case, you're going to be switching this guy right here. And so once you switch that one out right there, notice instead of arginine, you get now histidine. This is a missense mutation. So you notice you get a change in amino acid. So instead of arginine, that could be a problem, right? If that histidine was critical for your function, then that could be a problem. Missense mutation, which is the other one. This one's going to give you a premature stop, right? So for instance, now you've got UAA, so you get to methionine, proline, and then you hit this stop codon, and then that truncates the protein. So this gives you a truncated protein. These are basically single base pair mutations. Now to get even worse, we get the frame shifts, right? And we already talked about frame shifts, didn't we? Because frame shifts are either going to be deletions or additions of either a single or two base pairs, right? And we saw how it shifted the frame, right? When we talked about the three-letter word, discovering the three-letter word. So I don't want to recapitulate that discussion because we already have a pretty good sense of how it shifts the frame in reading. But that would be a mutation. These frame shifts are really lethal. These are very lethal mutations, very nasty customers. They're almost associated, they're very aggressive in nature. So you don't really have a lot of hope in this one, right? The nice thing about missense mutations is that depending on where they happen, they can be somewhat permissive to function, right? So if they have all that big of a deal, your protein may still have a function, residual function, and you dodge a bullet on that one. But typically a frame shift mutation is nothing catastrophic, like disruption of the code that it's very aggressive, very lethal mutations. And you break down just how you're actually producing a functional polypeptide in the first place. The other thing that they kind of throw in there is what's called triplet repeat mutations. It's a group of mutations actually that are associated with this phenomenon where we see in some genes you have these triplets like CGC, but you have like it's like a repeat of CGC, CGC, CGC. And so what happens is in some situations, we know that these areas of repeats can expand. That is, as they go through mitosis or meiosis rather, they can actually expand, they can duplicate. So they can go from, say, for instance, 10 repeats to 20 repeats to 40 repeats. So they can grow and expand. And typically speaking, the more they expand, the more they kind of blow up the gene, which causes problems for the gene. And so as a result, we have disorders that are associated with these expanding triplet repeat areas. And Huntington's disease is one of those. It's a profoundly devastating neurological disorder. Huntington's disease is, it's a neurodegenerative disorder that's associated with expansion. So at a certain number of repeats, it's basically, subdiagnostic. So you don't have Huntington's. But then as you go from one family member to the next, then that site expands and then you become borderline. And then the next generation will expand and then it gets to full-blown hunting because it blew up the gene. And so we noticed this about some of these, and those result in this kind of blow up, if you will, of the gene itself. So we have like entire chapters. There's actually 2 chapters in the genetics class that we cover when we cover mutations. We cover small mutations in sequence, alterations in sequence, and then we cover big mutations, like alterations of chromosomes. And so it has its own chapter. So there's a lot going on there. So the chromosomal mutations are essentially the same things, only instead of happening in individual base pairs or small pieces of base pairs, small pieces of sequence, it's happening globally across the entire chromosome. So for instance, you still have deletions, but it's of the entire chromosome, right? You still have duplication, but it's of the entire chromosome or entire regions of the chromosome. Another one that we haven't seen is inversions. An inversion literally is where you cut out a piece of your chromosome, you pop it out, and you spin it around 180 degrees, and you plop it back in. So you invert it. So like, for instance, if it's going this direction, you spin it around, it's now going this direction. So you literally invert it. And a translocation is basically where 2 chromosomes swap parts of each other. It's called a reciprocal translocation. This is what it looks like. Here's what a chromosomal deletion looks like. We're going to take a look at the chromosome A, B, C, D, E, F, G, H, I, J. So here's your long arm, here's your short arm, this is your centromere there. So what we're going to do is we're going to delete out the ABC segment. And so what we get in our deletion chromosome is A with the omission of our BCD. And then we have E, F, G, H, I. So you can see you've omitted that entire region. Now notice, these are regions, not genes. So we're not talking about single genes that are being deleted. We're talking about regions of the chromosome that are being deleted. Any region could have hundreds to thousands of genes in them. So when you're omitting an entire region of a chromosome, you're looking at potentially a very catastrophic alteration in the chromosome for a deletion. Now, what happens if you duplicate it? So we're going to take the same scenario, the same area, so the BCD version, and we're going to duplicate this. So we're going to go A, BCD, and then BCD again. It's like a stutter, right? And then E, F, G, H, I, J. So notice here, you basically duplicated that BCD region. Again, also, not just regions, but many, many, many genes within those regions, right? So you're talking about a ton of potential genes that you could have just duplicated here. And considering that we don't tolerate extra DNA very well, we don't tolerate extra chromosomes very well, then this could be for us a catastrophic fail. Okay. Now, what about an inversion? Let's take a look at the inversion. So we're going to take a look at BCD again. In this case, we're going to cut it out, and we're going to spin it around 180 degrees, and we're going to plop it back in. Okay, so we're going to take our BCD, and we're going to basically pull this out, spin it around, and notice now it's going to be DCB. So notice what we did, we spun it around. It's now backwards. But actually, this is inaccurate because a true inversion, this isn't a true inversion. A true inversion should look more like this. DCB. That's what the actual inversion should look like, right? Because everything is moving in that direction now, where everything before was read in that direction. It doesn't change the sequence in there. It just turns it around, right? Which is the reason why sometimes inversions are not catastrophic because of the inversion itself. But what it's catastrophic for is because of the breakpoints. If the breakpoints of the inversions are in very bad positions, like if you're breaking off the promoter region of a cancer gene and putting it next to a skin gene or something like that, then you're basically creating a different regulatory area in new places. So you're creating these breakpoints and they're putting them in new neighborhoods, and they may not work very well. We've actually seen that. As a matter of fact, that's how we discovered some of these inversions, is by their dysregulation of gene control, is because you've got a different regulatory region in front of a gene that didn't have that before, and it's not correct. It's causing problems. That same breakpoint idea we see in translocation. So in translocation, basically what you have is a swapping of DNA between 2 chromosomes. So here we have 2 chromosomes, A, B, C, D, E, F, G, H, I, J, and we have K, L, M, N, O, P, Q, R. what's going to happen is we're going to break off these two pieces and swap them. So the KLM will go to this guy and the ABC will go to this guy. So here you can see you've got KLM attached to DEFGHIJ and you have ABC attached to NOPQR. Again, not necessarily is it catastrophic that you have KLM over there, right? You still have it. You didn't duplicate it, right? You didn't delete it. It's still there. So why is the location a big deal? Well, technically speaking, the location isn't a big deal. But what makes it a big deal is the breakpoints. And actually, this is how same thing is true for the inversions, right? It's the breakpoint. So when you have a regulatory region associated with the M region that's now in control of D, then you're going to have a misregulation and a misexpression of D. That's actually how we found translocations, is because we were studying leukemia. Approximately, we're trying to figure out, well, how is this working exactly? And what we noticed was it was actually familial leukemia is what it was, so which is rare. And what we noticed was that there was a patient population in Philadelphia where a family had this sort of familial leukemia. We were studying it and we're trying to figure it out. We called it the Philadelphia chromosome. What we noticed was this familial pattern was caused by a translocation, where you had the breakpoints were unfavorable, where a regulatory region was actually dropped behind a cell division gene that promoted cancer. So literally that new regulatory region from the translocation started misregulating a cell division gene, which then caused cancer progression. And then that's what caused it. So we realized that it was not necessarily that there was anything wrong with the sequence. We couldn't figure out, like, well, what mutation is segregating with this disease type? We couldn't figure that out. What we noticed was it wasn't necessarily a mutation in a gene. It was basically that swapping of the DNA created a misposition of regulation and caused dysregulation of these cancer promoting genes that cause the leukemia, the familial leukemia in this family. Well, cancer cells are a train wreck. Yeah, cancer cells are basically like a genetic nuclear bomb. And so the thing is, normally, like if we get too much of this damage, it'll become catastrophic for us and we'll trigger like a cell suicide type of a mechanism. And so we'll die. But cancer cells don't. So they don't die. They just keep going and they keep perpetuating this. And so eventually some of those breakpoints are going to give them advantageous outcomes. Like if they can get a breakpoint that now starts to turn on telomerase, for instance, then they've got cell division immortality, right? If they can create mutations or breakpoints, however, whether chromosomally or sequence-wise, that turns on blood cell development, angiogenesis, then now they can grow without bound. So, and that's kind of and so there's no consequence to that because there's nobody around anymore because they killed P53 to tell them to stop and to die. So normally these are bad things for us, right? And so obviously these are all different types of mutations. Like I said, in the genetics class, which I'm all sure you guys are fired up to sign up for, all like 2 of you are actually in there, which means I'm not gonna have a job in the fall. But anyway, I will. Don't worry about me. I've got my cardboard sign ready, so don't worry about me. I'm just kidding. Yeah, they probably make more money than I do. Unfortunately, that's probably, and especially like the 3 kids. I could really juice this one up. I could have the 3 kids up there. I'd be like, yes, that's what I'm saying. You got to know how to manipulate people. That's mostly what street corners are about. But anyway, just saying, was that a little cynical? That was a little bit. Okay, we'll just keep going. Right, but basically these are mutations. Now remember, the reason why I bring up the genetics class is because in the genetics class, remember I said what we do is we take kind of the genetics chapter 12, and then we start to broaden and extend from there. Well, one of those big extensions is 2 chapters actually in that class where we talk about mutations. So there's an entire chapter just on the micro mutations, the sequence, the missense mutations, and things of that nature. And then we have another chapter on chromosomal mutations where we talk about things like non-disjunction and errors in meiosis and aneuploidy and and things like this, translocations and inversions and things of that nature, right? So mutations essentially are a big deal because they happen a lot. And in addition, mutation is one of the major mechanisms of evolution, and that's true. But I've already made my thoughts pretty loud and clear on the limitations of mutation, right? Which is one of the problems, because not only do we have mutation, and this is just to say that mutation is problematic, obviously, right? Because when you blow up a chromosome, bad things happen. It's usually, nobody has ever observed actually a good thing happen when you blow up a chromosome. But we've seen a lot of disaster when you blow it up, bad things happen, right? We call that medicine. And there's a lot of people out there with genetic disorders of one stripe or another, and none of them are good, right? There's not a single genetic disorder out there that everybody's like, ooh, I want to sign up for that one. That's good, right? Every single one of them are bad. Let that sink in for a second. And then on the heels of that observation, we have things like, oh, but mutations are the starting point for evolution. Oh, great. So you're saying that natural selection is going to omit me from the population because I've got a genetic disorder. That's basically how that works. And that's the problem, right? Because when you take a look at it, I mean, obviously evolution is a genetic story, but we have to actually be serious about what some of the limitations are of that, right? Because when you take a look at most evolutionary studies, a lot of them are actually happening. Most of us aren't really studying evolution like way back 3-4 billion years ago, because a lot of us realize how impossible that is, because there's just too much time elapsed and there's too many variables and there's too uncontrollable and you can't really get access to it. Most evolution is probably done, to be quite honest with you, from about the taxonomically, from about the family level forward. Now, take that for a second. Let's just sit there for a second. If a lot of evolutionary studies are taken from the family level forward, or maybe the class or order family forward, then what that means is, let's just draw a line there. Let's just imagine, forget about everything else, right? Forget about everything else. Let's just start with that. What we have in that pot is a giant pot of genetic variation that's already there. So then, when we're trying to figure out how families evolve into different genuses and into different species, right? We have two really major mechanisms going on. One is variation, not new variation, right? But pre-existing variation. And I'm just saying it's there, right? What I'm doing is I'm drawing a line. I'm not saying like, it just kind of came out of nowhere. I'm just saying that when we start our studies, we're kind of usually starting at a level, and so we're already have this armful of variation. That's not new, right? If you're starting from there and going forward, you have an existing amount of variation. But from that point forward, so from family forward, you have two mechanisms working. You have mutation, which gives you new variation. And you have adaptation, which is old variation. And in reality, evolutionary biologists are so obsessed with mutation, and I believe that they should try to broaden themselves a little bit and kind of expand a little bit. But when they're working at like the family level, oftentimes, and this is how UC Davis was, right? Because they were saying like, okay, so here we're going to do a phylogeny. We're going to basically draw this phylogeny. We're going to start off with class. That's where we're going to start. And we're going to take this class of organisms and we're going to draw a phylogeny of them. And the idea was that whenever you have a phylogenetic screen, right, of all these organisms that are kind of branching off from each other, was that this is your common ancestor. And then these guys continued on, but there was a mutation that we symbolized by a hash mark that happened that caused this branch to branch off from it. And then, of course, there was a mutation there that caused that one to branch off from it. And then, of course, as you get like multiple branches like this, then there's a mutation that branched off of that one. this one got another one, this one got another one, this one got another one, and so forth. And then what you have down here is species. But the idea is they're starting from maybe family up here. But here's the problem. This is basically, by the way, don't get too wadded up in that one if you didn't make any sense of that at all. That's hardcore evolutionary phylogeny. I'm just saying you how we traditionally think, how the Darwinian model thinks, is that all current variation is basically new variation that comes by way of mutation. But I would argue that from the family level, you've got a combination of old variation that's already come from 0 to there, right? Because we have an entire history going from domain all the way up to family. So we've got some variation that's accumulated since then. That's already there in the pot. So that's what I'm calling pre-existing variation. And then from family forward, mutation or new variation. But here's the problem. What Darwinian phylogenists say is that these changes from family to genus to species is associated with mutation. But the problem is, like in most likelihood, it's actually more just adaptation of existing variation or a combination of the two. Okay. Most Darwinians don't like the latter argument. But the problem is there's already some titanic problems with supposing that, okay, we're asking for a mutation here, and then a mutation here, and then one here, and one here, and one here, and one here. And wait a minute. How many good mutations have I seen in my lifetime? None. So how long are we waiting for these mutations to occur? Do we even have that much time in what we think is Earth history? So obviously there's got to be something else going on here. I'm not saying that mutation isn't a thing, but it is so catastrophically and titanically negative that it doesn't actually support what we actually see. There has to be something else going on. And that's basically what I'm saying, is that typically, like, especially when you're looking from family onward, you're looking more at like existing old variation that then gets sort of fine-tuned and chiseled away through adaptation to the environment. Okay, it's not unlike what we did to dogs, right? You took basically pre-existing variation that is in a common dog, and you adapted that to a teacup poodle, which you can put in your hand. And I know you can do that because I've actually done that. My dad had a teacup poodle. He's a little guy, cute but little. Or a mastiff, which one of my A&P students has next time, and he's like a massive guy, right? And everything in between. Now, were there mutations involved in that? In some cases, yes. Like the little smash face dogs, that represents a mutation. But is there a mutation, say for instance, between a beagle, who's a smell dog, right? And a German Shepherd? No, that's natural variation. That's adaptation of natural variation. So what we realize is when you have this massive reservoir of variation because of the way genetics is geared and you're sexually reproducing, that means that you have a capacity to generate a lot of different organisms that essentially we would classify as different species. That makes sense? So the common ancestor didn't give rise to multiple species through mutation. The common ancestor gave rise to multiple species simply by what they had genetically being adapted and fine-tuned to very different environments. Right? The big bear population, a good example, right? I mean, the grizzly bear, polar bear, black bear, right? So they're all interbreedable. Their common ancestor was basically, they could hybridize between all of them, which basically means they're all still related. So what you did was basically take bareness and adapt it to specific environments. But they're not so far along that they become reproductively isolated. So you can see how mutation is a thing, but there's a lot more to the story genetically. But Darwinian people tend not to think about that. We're just now starting to get there. They're kind of coming around. So I don't want to beat them up too badly.
So what's the difference with polar bear and a grizzly bear are different species, but a beagle and a German shepherd are the same species?
Yeah. Well, that's another thing that complicates this entire thing, which is one of the reasons why I'm not a big fan of the whole Darwinian the morphological phylogeny approach, which is usually what they do, is because how do you define species? A lot of people don't know that there's actually 6 different definitions for species. And depending on which definition you use, it'll shade your conclusions. So defining what a species is actually not as straightforward as we think. Which is the reason why we, have to be honest, intellectually honest with kind of how we approach things, especially in evolution. I'm not saying we don't do it, but we have to consider things. Like when I went to UC Davis, they did talk about adaptive radiation, but if I were to sit there and tell them, it's like, listen, I honestly think that speciation from the family level down is mostly adaptive radiation of pre-existing variation and not really mutation, they would have kicked me out of the university. That would be like heresy. That'd be heretical. But the problem is we see it all the time. We can see it in the bear population, right? So a lot of that is, and that's very strong because, it doesn't violate natural selection. It doesn't disregard mutation. Mutation's there. But in some cases, it's like, depending on where you're starting from, you could be looking at more like adaptive radiation of existing variation, and you could get a lot done. Right. I think if somebody were to take a look at a teacup poodle and a mastiff, they would naturally, like if we did the morphological thing, if we didn't know that they were the same thing, if we didn't do that ourselves, the way we classify species is we probably put them in 2 different species. But the problem is they're not the same species, are they? They're not different species, are they? But that's exactly what we do with other organisms. We take a look at them, we're like, oh, hey, you're fundamentally different, but we're going to put you in a different species set. But it's like, wait a minute. The same logic you use there, you could use against every single breed of dog. And instead of breed, you would create a separate species for the poodle and a subspecies for the teacup versus the toy versus the standard. So those would be a subspecies. That's morphologically, that's how most of the 19th century and the early 20th century, that's how they did evolution. Is they basically looked at stuff and said, oh, hey, you look pretty similar. So phylogenetically, we're going to put you in the same category. I don't necessarily think that's valid. But anyway, that's the semester. Go on. You like that one? You like that? You're like, I don't need you anymore. No. You like that? Yeah.